Start? Okay. 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 So yeah, welcome to the second lecture today. So what I want to start by doing today is to make things a little bit more visual. So I think yesterday I explained to you this concept of fitness landscapes, and today I want to begin with showing you some pictures, and later on I'll also show you a lot of experimental examples. So let me just recapitulate what I explained to you yesterday. So the way I've introduced these fitness landscapes is essentially as functions on a space of binary sequences. So we describe these genotypes by binary sequences. So we had these functions, and I had these two versions, the sigmas and the taus, that I will get back to. So sigma and tau are binary sequences. Of length L. And they are binary because we only keep track of whether we have a wild type or a mutant at a certain locus. And the sigmas are sequences of -1s and 1s. And the taus are sequences of 0s and 1s, right? But for the present discussion, this actually doesn't play a big role. So these things form a sequence space. Now, for something to be a space, you need a notion of distance, okay? And here there is a natural distance measure, which is called the Hamming distance. The Hamming distance. And this, I think, is familiar to anyone who has ever done a sequence alignment. So the Hamming distance is just the number of entries at which the two sequences differ, right? So this is the number of different entries. And I'm going to call this, so for two sequences, I will denote this by d of sigma, sigma prime. So this is the Hamming distance between the two sequences. And this can be, if the two sequences are the same, this is 0. The maximum it can be is L, because then they differ in all elements. So this is a number that can be up to L. Okay. Now that we have this space, we can draw some pictures. So let's first look at the case. Again, the case of two loci, which I discussed extensively yesterday, and I'm going actually now for I think for most of this, I'm actually going to use the tau representation again. For some reason, I find that more intuitive, so I'm going to consider 0s and 1s, right? And so then we have the sequences that we have are 0, 0. 0, 1, 1, 0, and 1, 1. Okay, so these are all the possibilities. And now we can make this into a graph by drawing a line between sequences when they are at distance 1. Okay, so we say that there's a link between, so we call this, and this will be called then a Hamming graph. We connect sequences at distance 1. Okay, and so here this gives us this little square, right? So these are connected. And so you see that in this case of two loci, the sequence space is just a square, okay? Okay, now we can do the same for three loci. And I'm going to draw this in a kind of suggestive way. So what do we have here? So here I have, say, the wild type of all zeros. Then I go in one direction. Here I have one type of mutant. Up here, I have maybe another type of mutant. But then I need to introduce a third direction because we have now three entries, so we need actually 3 dimensions. So we need to draw another axis here. Okay. And then you see how this will work. So now we can sort of connect things. So up here we will have 1, 0, 1. Okay, and then we need to go, so now I'm getting into trouble here. If we add another one up here, then we get to 1, 0, 0. Okay, is this getting right? Yeah, so these two are also connected, right? And then we need somewhere the all ones, which is connected to these. And then something is missing. Yes, we have something down here, right? So here there should be the 1, 1, 0, I guess, right? Okay, it's a little bit squished, but you can see what it is. It's a cube, okay? Okay, so for L equal to 3, we get a cube. And so for general L, these things are actually called hypercubes. Okay, so for general L, these sequence spaces are hypercubes. Of dimension L. Okay, and obviously this becomes sort of harder to visualize as you increase L, and this will actually be a theme that we'll encounter today. Also, when you look at empirical data, it's not so obvious how to visualize these things. But so let me now show a couple of pictures. So this is now a slide showing these hypercubes. So here there's even the one locus hypercube, which is just a line. Okay, so here we have the square, the cube. This is now the 4-dimensional one. There are sort of various ways of representing this. One way is to sort of put 2 cubes and to connect them. This is a 6-dimensional one. And what you can sort of see is that these things become very highly connected. And the reason is that the largest distance that you can go from one end to the other, so from all zeros to all ones, the largest distance is always L. Right? So the maximum Hamming distance is L. This is also called the diameter of the space. At the same time, there are 2 to the L sequences, so you have something which has exponentially many sequences, but it has only a diameter of L. So this becomes very highly connected, and there are sort of things to figure out about the geometry of this space. And if you're interested in this, so one of the exercises, actually the first exercise that I put into this problem set, so problem 1, is about the geometry of these spaces. Okay. Geometry of Hamming spaces. And in fact, in that problem, this is sort of generalized. I mean, so here, throughout or most of this lecture, except for one empirical example, I will be focusing on this binary case. Now, of course, if you think about DNA sequences, for example, you can have 4 different alleles per site, or if you think about proteins, you can have 20 different alleles per site. And in principle, you can generalize this concept to sequences that have entries from alphabets that are larger than 2. And these things are then still called Hamming spaces. In this problem, I'm asking for some properties also for general alphabets. Okay, general alphabets. But for today, we'll be mostly talking about the binary case. Okay, so this is sort of the space on which these fitness landscapes are defined. So in some sense, fitness landscapes are just functions on the sequence space. Now, before going on, I want to do a little bit of history. Okay, so as I mentioned yesterday, this concept of fitness landscapes was introduced by Wright in 1932 in a publication. And interestingly, in that publication, you can already find these hypercubes. Okay, so this is this famous paper. The title is The Roles of Mutation, Inbreeding, Crossbreeding, and Selection in Evolution. And here you can see exactly the way Wright represents these things. He doesn't write zeros or ones, but he considers here a wild type, and he has 2 mutations A and B, and here we have the double mutant. Here we have 3 mutations A, B, C, and so on. Okay, so you see that this is exactly the same geometry. Now, in this paper, there's also a second picture, and the legend has it that this second picture was introduced at the urging of the editor of this paper, because he said, well, this is somehow too abstract, biologists will not be able to understand this, so you have to somehow make it more intuitive, okay? So Wright put into here the second picture. What is the second picture? The second picture is a topographic map in 2 dimensions. So Wright's idea, of course, was that you should now assign fitness values, just as we have done here. You should assign fitness values to all these combinations. And so you have a function defined on this space. Now, to draw this function is not so easy, right? I mean, how do you, we'll see that later, how do you actually, visualize a number that is attached to each of these places? It's much easier to do that in the 2-dimensional plane, so this is what he did. So this is now supposed to be a 2-dimensional Euclidean plane with a function defined on top of it, and it's a topographic map, so these pluses here are peaks, and these minuses are valleys. Okay, so this is now Wright's representation of the fitness landscape, but he writes in the paper that this is actually an inadequate representation of what he's trying to show, because obviously a 2-dimensional plane is something very different from a hypercube. Okay. Nevertheless, this is in some sense the image that really stuck. So if you open up a textbook on evolutionary biology, and there's some discussion of adaptive landscapes, typically you will see pictures like this, or maybe 3-dimensional renderings with peaks and valleys and so on. And for this reason, mainly, I think, for a long time, this concept of fitness landscapes has been considered to be more of a metaphor. So it's something that you can use to think about how evolution occurs, how populations sort of move up in fitness landscapes, but it has never been, or for a long time, it wasn't taken seriously as an actual way to represent real things. And I think this has now changed, and part of the reason is that this kind of more proper representation is becoming more common, as we'll see later in the empirical examples. But let me first go on a little bit with a history. So what was sort of Wright's motivation to think about this? So his intuition was that the complexity of organisms implies that in a typical fitness landscapes there will be many peaks. So there will be sort of many different solutions of different quality to the same kind of problem posed by the environment. And so he writes here in this paper that in a rugged field of this character, so with this he means the landscape, selection will easily carry the species to the nearest peak, but there will be innumerable other peaks that will be higher, but which are separated by valleys. The problem of evolution, as I see it, is that of a mechanism by which the species may continually find its way from lower to higher peaks in such a field. So he conceived this as a kind of obstacle to evolution, right? So he said, okay, evolution will find sort of suboptimal solutions, but to actually find the best solutions, you have to find some way of crossing the valleys. And this was actually the rationale for him to invent the shifting balance theory with a coupling between geography and environmental change and so on, which is a rather elaborate scenario, which I think generally nowadays people think that this is not very realistic because it requires too many things to happen in a certain order. Now, whether this is really a problem for evolution or not is actually a question that is being debated up to this day. I'll get to that in a minute, but before getting to that, let me show you another founding paper by Haldane from 1931, where he essentially also introduces the fitness landscape also, and these words essentially are the same as the fitness landscape picture that I showed you. So he considers M genes, so this is what I call L. Any population can be represented by a point in M-dimensional space. All populations being represented by the points of a regular orthotope or hypercube. So here actually the word hypercube already appears. Each of the 2 to the m apices of this figure represents a homozygous population. So these are the vertices of this cube, and each of those is like a pure genotype. clearly the condition for stability of any such population is that no change in a single factor should yield a more viable type. So what does he mean by that? This means that if I'm, for example, sitting at 1, 1, 1, then this will be a stable situation if any mutation to one of the neighbors lowers my fitness. So this is essentially, and I'll formalize this a little bit later, this is what we call a fitness peak, right? So a local peak means a site which has larger fitness than all the others. In other words, no adjacent apices can both represent stable populations. The maximum number of stable populations is just 2 to the m-1. Why is that? Well, this is simply because if you want to try to have many peaks on this landscape, the best you can do is to say, suppose this one is a peak, then of course its neighbors cannot be peaks, right? But this one can, for example, be a peak, right? Again, its neighbors cannot be peaks, but this one can be a peak. And I guess there's a line missing here, actually. There should be a line here. Okay, and then there should be one more possible peak, which is probably, which one? This one, right? So this is the way you can get 4 peaks. And in fact, part of this problem here, problem 1, if you're interested, is to find what is this maximum number if you have an alphabet of more than 2 letters. Okay, so what is the maximum number of maxima that you can accommodate on a multi-allelic space? Okay, so this was Haldane. So he also talks about these peaks, but his interest here is more to think about how many peaks there could maximally be. But whether there are peaks at all and whether they are relevant, as I said, was debated already at this time. And there's a famous correspondence between Ronald Fisher and Sewell Wright about this. And here Fisher writes the following. So this is a kind of mathematical argument. He says, in one dimension, a curve gives a series of alternate maxima and minima, right? So if you draw any curve in one dimension, it has some complicated shape, the number of maxima and the number of minima will always be the same. But in 2 dimensions, 2 inequalities must be satisfied for a true maximum, because you have, if you remember your basic calculus class, for something to be a maximum of a 2-dimensional function, you need the matrix of second derivatives to have a definite sign, and these are 2 conditions. And so he says, well, about only 1/4 of the stationary points will satisfy both. And roughly, I would guess that with n factors, so n dimensions, only 2 to the minus n of the stationary points would be stable for all types of displacements. So essentially what he's saying here is that in high-dimensional fitness landscapes, most of the points are not maxima or minima, but they are saddle points. And I think this may be something that Daniel will be. Are you going to talk about this? The complex fitness landscapes in high dimensions, maybe? Okay, but well, Daniel has interesting things to say about that. So basically, he says that as you go on, there will be less and less maxima, right? So, and this suggests that true stability in the case of many interacting genes may be of rare occurrence, though its consequence when it does occur is especially interesting and important. So basically, his argument is that because of the high dimensionality, somehow there will not be any peaks, or very few, and they will not be relevant. And we'll see in the mathematical theory probably tomorrow to what extent this is true or not. I think my view now is that in some sense both Fisher and Wright have a point, but this 2 to the minus n, the rate at which the frequency of maxima decreases with dimension actually is overestimated by this argument. Okay, and the last historical remark, so there's a very influential paper by Maynard Smith, who sort of transported this picture to protein space. Of course, at the time when Wright and Fisher devised these ideas, they didn't know about DNA and the structure of proteins. So Maynard Smith sort of brought this picture into the new era. So he thought about the space of all proteins, which would be in our language a Hamming space with 20 possible letters at each side. And he thinks about protein evolution, and he says it can be understood by analogy with a popular word game. The object of the game is to pass from one word to another of the same length by changing one letter at a time. So this essentially one distance moves, right, Hamming distance 1, with the requirement that all the intermediate words are meaningful in the same language. So this means that if you want to mutate a protein, if you want to go from one state to the other, you probably want to do it in such a way that all the intermediates are also functional. Okay? And so the nice example here is to convert the word into the word gene, and he does that in these steps. And so this directs the attention to what we nowadays call mutational pathways, so ways in which you can move in these fitness landscapes in such a way that a certain function is preserved or maybe even improved. Okay, and this is something that I, again, will get to a little bit later. Okay? So this is what I wanted to say about the historical part. So now I think we can, in principle, okay, where did the guy go? We could turn this off again. Or are there any questions about this? Okay. All right. So let me try the new technology for wiping the blackboard. Okay. So let me now go on with the development of the theory for a little while before I get to the empirical examples, because we need a few more concepts to be able to understand what is going on in these experiments. And so let me just recapitulate what I did yesterday. We had these, what I call the Taylor and Fourier representations. And I think maybe just to clarify this a little bit more, so both of these, so if you let me write them down again in the way that I did this yesterday. So here we distinguished between the first order effects, selection coefficient, and then here we had all these higher order effects, right? And here we sum over subsets. And the second representation has the A coefficients. What I want to emphasize that both of these are actually, what they actually do is that they are linear transformations between the fitness values and the coefficients. Okay, so these are linear transformations. And to make this, slightly more concrete, I thought I should maybe simply write down the matrices for the case L equal to 2. Right, so if I want to reconstruct my fitness values, so there are 4 types in this case. So I want to reconstruct my fitness values from the coefficients. What do I have to do? Well, so okay, so on this side I have my coefficients, right? So the coefficients in this case are F0, F1, S1, S2, and what I called epsilon 1, 2. And so F0, 0 is simply F0, right? So the first entry in the first row of this matrix, I just have 1, 1, and all the others are zeros. F1 is obtained by F1, F0 plus S1. Okay, so here I have this row. F01 is the sum of these two. Okay, so here we have 1, 0, 1, 0. And finally, the fitness of the double mutant is just the sum of all of them. Right? So this is my matrix. Okay, so this is the, and you can sort of find these matrices in more generality again in this paper by Polvik. And to sort of bring out the advantage of this Fourier representation, let me show you what the same thing would look like for the Fourier case. So then we have, let me just write the types here with minus, minus, and so on. So minus stands for minus 1, and plus stands for plus 1. So this is now related to my coefficients. So here this would be A0, A1, A2, and A12. And this matrix, so I have to look it up. But essentially what I said yesterday as well, and this is sort of clear from the structure here, that all these rows contains only 1s and minus 1s, right? Because the only values that this thing can take are minus 1s and 1s. And so the matrix that you actually get is the following. So up to some overall constant. So as I explained to you yesterday, F minus minus, so the zeros order coefficient is simply the sum over all the fitness values. Okay, so this is actually wrong. This is the inverse matrix, so let me do it right. So this is what you get. Minus 1, 1. 1, 1, minus 1, minus 1. 1, minus 1, 1, minus 1. And 1, 1, 1, 1. Okay, so this is what this matrix looks like in this case. And what you can sort of see by plain eye is that all the rows and columns of these matrices are mutually orthogonal. Okay, so this is what I mean, what I meant when I said yesterday this is an expansion in orthogonal eigenfunctions. So this is sort of what makes this transformation mathematically nice. And the inverse is simply the adjoint of that matrix. And so as a consequence, and this was sort of the last thing that I said yesterday, as a consequence of this, this allows you to decompose the fitness variance cleanly into different epistatic orders. So the consequence of this is that we can decompose into epistatic orders. So So this is just the variance of the fitness, so the fitness, the mean of the fitness squared minus the mean fitness squared. Okay. So if I need also a notation for averages. So if I use this to write the average over the landscape, right? So this is this thing, and so this now decomposes in the following way. So here again we have all the subsets, and here we have these squares of these coefficients. And so this allows you to sort of, if you just look at the k equal to 1 terms, then those gives you the non-epistatic part, and k greater than 1 gives you the epistatic part, right? So k equal to 1 is non-epistatic, and k greater than 1 is epistatic. And so with this, you can, if you want to know how much epistasis is there in my fitness landscape, you can represent that by just looking at the sum over k greater than 1, say divided by the whole sum, right? So that will give you the fraction of the variance that is actually related to epistasis. Okay? Okay, so this is essentially this kind of analysis that I introduced yesterday. Now the question is, you know, when I showed you these historical slides, what people of course worried about in terms of fitness landscapes from the very beginning was the presence of peaks, right? This is what Wright talks about, and this is sort of what the debate is about. And the problem with this representation, although it's a nice measure of epistasis, is that it doesn't actually tell you anything about peaks, right? So you cannot, from this, the only thing that you can tell from this quantity is that if all the terms of order k greater than 1 are 0, then there can only be one peak. This is sort of the only thing that you know. But beyond that, it doesn't actually tell you anything about the structure of the landscape as far as what is normally called ruggedness is concerned, okay? And for this, we need a kind of a different concept of epistasis. And this is the concept of sign epistasis. Okay, so this will be the next part, which I think in my notes probably has a number 3, because yesterday I did something that had a number 2. Okay, so this is now the concept of sign epistasis that we need to talk about. This is something rather natural, but surprisingly, it was introduced, as far as I know, only a bit more than 10 years ago. And this is really the content of the paper by Dan Weinreich, Watson, and Chow that I also gave you as reading material. Okay, so this is what I want to explain. And so the goal here is to develop epistasis measures that can tell us something about ruggedness or accessibility of the landscape. Develop epistasis measures that are informative ruggedness and accessibility. So accessibility meaning in the sense that I just mentioned in the context of this quotation from Maynard Smith, you know, how easy is it to get from one place to another on this landscape? Now let's again look at the case of two loci, okay? So for the case of two loci, we have this simple representation of the fitness landscape in terms of the selection coefficients and the epistasis, right? And again, I'm using the tau representation here. And now we'll ask the following question. So if I do the first mutation on the wild type background, its effect will be S1. Okay, but of course I can also do the first mutation on the background of the second one. And so what is the selection coefficient of mutation 2 or mutation 1 on the background of mutation 2? Okay, so let me call this S1 with a 2 upstairs. Okay, so this is now S1 on the background of S2. So what is it? Well, it is obviously the fitness of the double mutant minus the fitness of the mutant which only has the second mutation. And if you use this formula, what you see is that this is equal to S1 plus epsilon 1, 2. Okay? So it's modified by this epistasis epsilon 1, 2. Now what can happen, of course, so all of these things, of course, can have any sign, right? They can be positive or negative. In particular, it's very possible that epsilon 1, 2 and S1 have different signs, right? So epistasis can be negative for a beneficial mutation. And then it can happen that this thing actually is negative if S1 is positive and vice versa. Okay, and this is what sign epistasis means. So sign epistasis means that epistasis changes the sign of the mutation. So S1 and S1, 2 have different signs. Which means that S1 times S1, 2 is negative, right? So the product is negative. This is how you would test whether this is true or not. And now you could do the same test, of course, for the second mutation, and it could be sign epistatic or not. If it's also sign epistatic, then we say that there is reciprocal sign epistasis. Okay, reciprocal sign epistasis means that S1 times S1, 2 is negative and S2 times S2, 1 is also negative. And you can sort of work out what the conditions for epsilon 1, 2 are for this to be true. Okay, so what does this now mean? Again, it's important to sort of visualize this a little bit. And for this, I'm going to introduce a sort of modification of the hypercubes that I drew for you before. And I'm going to introduce something that we call fitness graphs. Okay, so let me write it maybe here. So to visualize this, so this is one way of representing fitness landscapes for many loci. As you'll see, it doesn't contain all the information, so it's not, it's not exhaustive, but it contains the information that is relevant for the present discussion. So let me explain to you what that means. So if we again look at the two locus case, let me be consistent. So this is our two locus situation. And now in order to make this into a fitness graph, we draw arrows in the direction of increasing fitness, right? So that's the idea. So there's a hypercube. It's mathematically speaking, it's an orientation of the hypercube, right? So you put arrows on the links. So it's an orientation of the hypercube. Okay, so we say here arrows point towards increasing fitness. So let's do that. So let's suppose that both of the mutations are beneficial. So this means that these arrows point in this direction. Okay. Now, how would I see in this picture whether there's sign epistasis? So if there's no sign epistasis, then this means that if I do the first mutation in the background of the second, this corresponds to this arrow, it would still be beneficial, right? So this means in the absence of sign epistasis, this arrow should point in this direction, and the same is true for this one. So you see what absence of sign epistasis means is that arrows that are parallel to each other, that point in the same direction, also have, or that are parallel to each other, point in the same direction, right? So no sign epistasis means that parallel arrows point in the same direction. Let me maybe just illustrate that also for the cube here to make this clear. So now here we have the 3-locus case, right? So this is now a cube. And so here, for example, this link would correspond to one particular mutation direction. And if there is no sign epistasis with respect to this mutation, it means that all these arrows also have to go up, right? So these are sort of the arrows that are parallel transported from the first arrow. So this would mean that there's sort of no sign epistasis at the third locus in this case. Okay, and now we have, of course, other scenarios as well. So we could, for example, imagine that that there is sign epistasis for the first mutation, which means that if I do the first mutation on the background of the second, the sign changes, so then this arrow would be in the other direction, but the second mutation is not sign epistatic, so then we have this kind of picture, okay? And this is sometimes called simple sign epistasis, because it only holds for one of the mutations, but not for both. But then I can also have the situation that the graph looks like this. So here I go up, and here I go down, and here I go to the right, and here I go to the left. And this would now correspond to reciprocal sign epistasis. Okay. And this can obviously be generalized to any number of loci. So even as you see here, for a larger fitness landscape, I can always ask, does this, when I look at this mutation on different backgrounds, which essentially means that I look at the way these arrows change as I move them around the plane, whether the sign is always consistent or not. Okay. Now this is the concept of sign epistasis, which was introduced in this paper. Now, of course, as I said, per se, whether this is true or not cannot really be read off from this Fourier representation. So of course you can have epistasis that is not sign epistasis, and to distinguish the two kinds, Weinreich and collaborators also introduced the notion of magnitude epistasis. So magnitude epistasis means that as you move around, say as you look at the selection coefficient over different backgrounds, its magnitude changes, but its sign does not, right? So this is which wouldn't be visible in this fitness graph, right? So the fitness graph only keeps track of the signs. So this is one remark that I want to make here. Epistasis that does not induce a sign change in this sense is called magnitude epistasis. And the second remark that yeah. Yes? Yeah? No, what you want to look at is the, so you want to look at the same mutation in different backgrounds, right? So which picture are you showing? The first one. Okay, so here we have the first mutation in the 0 background increases fitness, right? The first mutation in the 1 background increases fitness. And here the second mutation in the 0 background increases fitness, and the second mutation in the background of the first also increases fitness. So that's what I should compare. Right? So you don't, of course, you don't want to compare different mutations. I mean, different mutations, of course, can have different signs. So you want to look at the same mutation only across backgrounds. Okay? And this really means just geometrically that these arrows are parallel. Okay? Right, so this is called magnitude epistasis. The other thing that I wanted to emphasize here is that all of this discussion, and also what will follow, this discussion related to sign epistasis, it's really a property only of the rank orders of the fitness values, right? So you actually don't really need the numbers, if you will. You just need to know which fitness is greater than which other fitness, and this is sometimes called the ranked fitness landscape. So sign epistasis is a property of the ranked fitness landscape. So with that, I just mean I just order my fitness values according to their size, and then I just give, I say the largest one I call rank 1, the smallest one rank 2 to the L, and then I just order them, and then I just assign, instead of assigning fitness values, I just assign the ranks. This is the rank fitness landscape, and therefore all of this discussion is invariant under monotonic transformation. So I can take my fitness and perform some transformation that is monotonic to some other, for example, and this is of course in experiments often the case, that you're actually not really working with fitness, but you're working with some proxy of which you think it's probably related to fitness. And as long as you can argue that this proxy is monotonic, then these properties will not depend on the scale. Okay, so it's invariant under monotonic transformations. Okay, so this is a nice definition. The question is, why is it important? So what I promised to you here is that this should be sign epistasis should somehow be informative about features of these fitness landscapes that are relevant for the evolutionary process, in the sense that this was discussed, for example, by Wright. And so this is what I want to explain to you. And for that we need, again, a little bit more, a few more definitions. Okay. So what are the consequences of sign epistasis? So to explain that, I first need to sort of introduce two quantities that are related to ruggedness and accessibility, two measures. The first one is what is a fitness peak? So this is fairly obvious. Fitness peaks or maxima. Okay, so a fitness peak is simply a genotype which has a fitness that is larger than all of its neighbors. Okay, so this means that f of sigma or tau, maybe I should be consistent and call them tau for now, is greater than f of tau prime for all tau prime which are at distance 1. Okay. So the number of these fitness peaks is obviously a measure of ruggedness, and this is probably the colloquially most frequently used notion of ruggedness. And these fitness peaks are sinks on the fitness graph, right? So for example, if you look at this, this reciprocally sign epistatic landscape, you see that this actually has two, so these have one peak, right? So here, the only peak is this one, right? So a peak in this representation means that all the arrows are coming in, right? This one also has one peak, which is sitting here. But this one actually has two peaks, which are here and here, okay? So this has one peak, and these have two peaks. So from the fitness graph, you can immediately read off whether there's a peak or not. A somewhat more subtle concept was again introduced in this paper by Maynard and collaborators, which is the concept of selectively accessible pathways. So here the idea is that you move around in your space along the link. So you do one mutation at a time, and this defines a pathway. And you say that such a pathway is selectively accessible if fitness increases in each step. Okay, so this is sort of a picture that is really based on the low mutation regime that Daniel was talking about. If your population is mutating in a regime where essentially mutations appear and then they go to fixation and then you wait a long time, then the next one comes. So in such a regime, the only way in which you can actually evolve is by having mutations which are beneficial in each step, right? At least if the population is large enough so that deleterious mutations cannot fix. So a selectively accessible pathway is a sequence of adjacent genotypes so you go one step at a time. Such that fitness increases in each step. A further important distinction is, so these pathways, of course, can be of different lengths. So, for example, if you look at this three-locus landscape, if I want to go from all zeros to all ones, the shortest path that I can take contains 3 mutations, right? Of course, I have to do 1 mutation at a time, but in principle, there can also be longer paths, right? So I could also, for example, go a path that looks like this, right? This would also take me there, but this would be longer, okay? So one can distinguish between so-called direct paths. So a direct path is one that decreases the distance to the target in each step, where the number of steps is equal to the distance, and indirect paths are other ones. Yes?
So magnitude epistasis indicates that the value of the sign chain can be anything, right?
The value of the selection coefficient can be anything, but its sign doesn't change.
Yes, so it could also mean that that value could be 0.
Well, okay, so this is a very good question. I mean, I'm basically ignoring the possibility that selection coefficients are 0. Okay, this is of course very much, at par with the proponents of the neutral theory. But so essentially, I mean, the kind of data that I will be showing tomorrow is typically based on mutations that have some effect, right? So then you basically don't see really neutral mutations. Of course, it could be, again, relating to Daniel's lecture, it could in principle be, but somewhere a mutation coefficient becomes very small, such that this S times N is no longer large, and then you might worry that things happen. But so here I will basically ignore that complexity and just say, I assume all those fitness values are different, there are no neutral mutations, and any selection coefficient that is not 0 is relevant. Okay? Right. So we have to distinguish between direct path and indirect path. So direct path are those where the distance. So let me say this. Let's say this. We start from some initial genotype. Let me call it tau 0. Or tau initial, and we end up at some final genotype, and then a direct path is where the number of steps in the path is equal to the distance. And indirect paths are those which are longer. So indirect path, as you can see in this example, in this binary setting, indirect path imply that you actually have reversions, right? So you have, for example, in this case, you mutate the first locus and the second locus, and then you sort of reverse the second locus, right? So this is so the indirect steps are related to mutational reversions. Okay, so these are the definitions, and now we can formulate the main results from the Weinreich work, which sort of show that why this notion of sign epistasis is important here. Maybe I can just, okay, I'll leave it there. There's enough blackboard space. Okay, so then the following is true. Okay, so the first statement is, and so all of these statements essentially say that landscapes without sign epistasis are in a qualitative sense almost like additive landscapes. Okay, maybe I should write this here. So sign epistasis is really the important part of the epistasis. Similar to additive. Okay, so the first statement is that if there's no sign epistasis, then the landscape has only a single maximum. Single global peak. This is actually rather easy to understand. So if there's no sign epistasis, then this means that each locus has a selection coefficient of a fixed sign, right? So this means that one state of the locus has higher fitness than the other. And so this means in order to get to the global maximum, you just have to put every locus into its better state, and that's the only solution. Okay, so this is fairly clear. The second statement is about the selectively accessible path. And so again, on an additive landscape, if you're not at the peak, then you will have a certain fraction of mutations which are not in their good state. Okay, and you mutate them into the good state and then reach the peak. So this means that you can, and in every step the fitness will go up. So this means that on an additive landscape, all the direct paths from anywhere to the peak are accessible. And what Weinreich and collaborators prove is that this is essentially equivalent to having no sign epistasis. Okay. So in the absence of sign epistasis, all direct path, this is important, path to the global maximum, global peak are accessible. And the converse of the statement also holds, which means that, and vice versa, which means that if all the direct paths to the global peak are accessible, then there cannot be any sign epistasis. Now this converse statement to the first statement does not hold. So even if there is sign epistasis, there could just be a single peak. In fact, the little example that I showed you before, so I had this the two locus examples, right? So if we look at this one again. So here we had sign epistasis, but we have only a single peak, right? On the other hand, if we have reciprocal sign epistasis, then, as we argued before, we have two peaks, okay? And so in fact, and this is a result that was obtained a bit later by Polwijk et al. Reciprocal sign epistasis is a necessary condition for having multiple peaks. Okay, reciprocal. is necessary for multiple peaks. So this was proved by Polwijk et al. In 2011. And it's sort of exemplified by this, but you can actually prove it in full generality. And maybe since I won't really be able to get into the empirical landscapes today anyway, maybe I can show you the proof. It's really cute. I think it's a very nice proof. So let me explain it to you for this third statement. So proof of statement number 3. Okay, so what we have to prove is that if there are multiple peaks, then there must also be a reciprocal sign epistasis, right? This is sort of the logical equivalent of this statement. So let's suppose we have 2 peaks. Okay, assume 2 peaks. These can be at some distance, and there will be a number of paths connecting them. Okay. Okay, so let's say this is peak 1. And here is peak 2. And here is fitness, okay. And now because these are peaks, I know that the first step must go down, right? So this might be one path. You know, it does something in between, something complicated maybe. Could even go higher. Okay, so this is my path. Now obviously, every such path has a point of lowest fitness, right? And this is actually the only point where the fact that this path is connecting peaks actually comes in. If these were not peaks, then the path could be like this, and the lowest fitness could be at the end, right? But because it's a path connecting 2 peaks, the point of lowest fitness has to be somewhere in between. Okay, so this is the point of lowest fitness. Okay. So now there are many such paths. Each of these paths has a point of lowest fitness. Okay. Now, out of all these paths, I pick the one where the point of lowest fitness is highest. Okay, there must be such a path. There's a finite number of paths. So pick, and so this is sort of like, you know, if you, in the opposite analogy, if you're walking in the mountains, you want to find the saddle which has sort of the lowest height, and here we want to find the lowest fitness. Okay, so we pick the path. Where the lowest fitness has the highest value. So let me assume that the path that I have drawn for you here is that path. And so now on that path, there are 2 mutations which correspond to these 2 steps, right? The 2 steps that lead down to this and up again. Okay. So now what I'm going to do, and I'm just realizing in my mind that there is a slight loophole, so I have to check if I can close that. Which Comes back to Daniel's warning never to do algebra or proofs in public. Sorry? Maybe that's a good idea. Yeah. Okay. So let me just let me just give you the argument. Oh no. There's not. It's fine. It's fine. I just had a logical flaw in my brain. Okay. So so what we now do. So we have some mutation here. Let me call it mutation 1, which leads me down to this valley, and a mutation 2 that leads me up again. Okay. So these are particular substitutions that occur at that particular point. And now I construct a new path where the order of these two mutations is exchanged. Okay. So we exchange mutations. 1 and 2. Now, the only thing that will then happen is that this part of the path changes, right? Everything else just stays the same. And so the only thing, so now I have to replace this point here by a point of different fitness. However, because I've chosen the path which has the highest value, I know that the new path has to have a lower value at this point, so the new path will look like this. Okay, so the new path has to look like this. Okay, and now you see that we've actually generated reciprocal sign epistasis, because these loci here, so this is now the locus, so this was mutation 2, so now this is 2 and 1. And so if you now consider these 4 loci here, these 4 points, you see that they exactly display reciprocal sign epistasis. So this is sort of the basic reciprocal sign epistasis motif. If you know, so here, and maybe this is actually a good point to show a slide. Yep. Yes. No, but you know what I'm saying is that, you know, the way I've picked this particular pair, I know that the fitness of the intermediate has to be even lower, right? So this is why they actually, and this is exactly the point. So what you see here is now that, so I actually don't have to say anything more. So what you see here is now that 2 now goes down and 1 goes up. Okay, so we have reciprocal sign epistasis. So the signs of both of these mutations now have changed. Okay. So this proves that if there are multiple peaks, we must have reciprocal sign epistasis. Okay. And so now you can ask, is there a sufficient condition? So this is a necessary condition for multiple peaks. Is there a sufficient condition? And in fact there is, and this was shown by Christina Krona and collaborators. Maybe I can just write it here. So this is like, or is there some, no, there's some space over there. So, but it's not a very good condition. So I think there's still, you know, room for work on this problem, which is that if all the instances of sign epistasis are also reciprocal, then the landscape has multiple peaks. Okay, if there's only reciprocal sign epistasis, if all instances sign epistasis are reciprocal, there are multiple peaks. This was proved by Christina Krona and collaborators in 2013. And so the last thing that I now want to mention also goes actually back to work by Christina Krona, and this sort of refers to the third paper that I posted as among the reading material for this part of the lecture. And I just, you know, I think it's a very nice paper, so, you know, I recommend to look at it, but I can't really say much about it here. But what I just want to mention is that this concept of sign epistasis, of course, means, because as I emphasized, sign epistasis is a property of the ranked fitness landscape. So in order to identify sign epistasis, you only have to note ordering of the fitness values. And so what Krona and collaborators have shown in this recent eLife paper, or have sort of elaborated on, because what this means is that you can infer epistasis only from ordering of fitness values. So you actually don't need to know the magnitudes, right? So a consequence of this notion of sign epistasis is that fitness information on or fitness rank orders can be informative about epistasis. And this could have a certain practical importance. For example, if you do competition experiments, then the outcome, if you don't measure, if you're for some reason not able to measure fitness very accurately, at least you can tell which strain has higher fitness than which other. So you can sort of generate rank order information. And if you do have enough of that, then you can infer whether there is epistasis or not. So this is the idea of this paper, Krona et al. In eLife. And just so you can ask under what conditions does this work, and we can again look at the two locus case. So let's again look at the two locus case. So there we have these three basic situations, right? Up to rotations. I mean, you can sort of rotate these graphs, but there are sort of three basic types, right? With all arrows parallel, with one anti-parallel pair, and with two anti-parallel pairs. So which of these three graphs imply epistasis? Which of these graphs can you sort of conclude that there must be epistasis? Yeah. The second and the third, right? So these are informative, right? These are informative of epistasis. And this one is not informative because it's consistent with just having additive fitnesses, right? Now, of course, each of these three classes contain several rank orders. So we have 4 genotypes. So there are 24 different rank orders. And if you look into the paper by Christina Krona, she has sort of listed all of them. And it turns out, and this is not difficult to check, that each of these graphs correspond to 8 different to 8 rank orders, right? So this one has 8 rank orders. This one has 8 orders, and this one has 8 orders, making altogether 24. And so this means that in this case, 2/3 of all rank orders contain information about epistasis, right? Are informative. And now you can ask, how does this, what happens to this if you go to larger sequence length? And it turns out, for example, for 3 loci, the corresponding number is already very close to 1. So for 3 loci, the combinatorics become sort of more cumbersome, but you can work it out. And so for 3 loci, the fraction of rank orders that are not informative is only 1 by 105. Okay. Now, of course, in an actual experiment, you will probably not have all rank orders, a complete rank order, because you might not want to compete everybody against everybody else, but you'll have some partial orders. And in this paper, you can also find information, and I think there's also some software that basically tells you how to, what kind of inferences you can do on the basis of these rank orders. Okay, I think this is a good time to stop. My time is almost over, so we can have some questions. Yes.
The single mutants have to have a defined rank order then? Or can 2 mutations have the same rank?
No. Again, I'm assuming here that there is a well-defined rank order, so all the fitness values can be ordered.
Yes.
Right. So this means that no 2, if you had 2 fitness values, right. Of course, for the fitness graph, this might not. If the 2 fitness values that have the same fitness are not adjacent to each other, fitness graph would still be well defined, right? So you could make, and this is sort of what Cronite are what they call partial orders. So if you have some that have the same order, then your knowledge is sort of incomplete, and you have some partial orders, but you don't have the complete rank order, right? And this is, of course, often, so essentially, what the fitness graph contains is a partial order which contains only information about adjacent genotypes, right? But that as such, of course, already allows you to say whether there is epistasis or not. Yeah. Yes. Can you say? Mm-hmm. Right. You mean from like the analog of this? So this L equal to 3. I'm not sure that there is a number for general L. I would have to look up the paper. No, you can see that this fraction that you can throw away goes to 0 very quickly. But then, of course, what they also do in this paper is to specifically, for example, you can ask, because, as I mentioned yesterday, there is, for some reason, people are very much interested in high order epistasis. So you can ask, what rank orders tell you that there is, for example, epistasis of the highest order? Okay, and then there is some condition for that. And I think actually I wrote down that number as well. So there's a formula for general L. So what they say is that the fraction of rank orders that imply L order epistasis is 2 over 2 to the L minus 1 plus 1. So this also goes to 0 as L becomes larger. It's fairly sizable for small L, but this tells you that if you want to infer something about specific epistasis coefficients, this becomes harder and harder as L becomes bigger. No, there's no assumption about the landscape. No. It's just, Yeah, this is not, this is not really, I think the main thrust of this is not with regard to models that can be correlated or uncorrelated, but actually with regard to data, right? So the idea is you give me some fitness values, or you give me information about rank orders, and I can tell you what kind of epistasis there is. No, this is again just the combinatorics of, the fitness landscapes.
The whole thing about talking, what order things are, is pretty sort of sweet at some point. That references it acts as if there's a bunch of different things you're adding together.
Well, if you compete two strains against each other, one will be fitter than the other, right? Yeah, I mean, I'm not.
How many coefficients do you need to represent it?
Yeah, I must. I mean, this sort of became clear to me yesterday also that I can't. I mean, there is a strong interest in higher order epistasis at the moment, but somebody, one of the students was asking me yesterday why is that so important, and I couldn't really give a good answer. I mean, it's just it's sort of. I think it's sort of more like again, noticing that there are sort of certain prejudices that people thought. If there are interactions, just like in physics, if there are interactions, you assume they are somehow pairwise, right? And right, exactly. No, I agree. I mean, there's no reason to assume that here, but it's. Yeah. I mean, in some sense, all of this, and this, I think this is what you'll see tomorrow when I show you the empirical examples. All of this is a kind of attempt to find ways of quantifying these landscapes and also to be able to then distinguish them, right? I mean, so one part tomorrow will be about comparing landscapes built out of mutations that have been obtained, that have been selected under different criteria, right? So, for example, you can select mutations that are individually beneficial, you can select mutations that are collectively beneficial, and so on. And you want to know, in what sense are the landscapes that come out of such an exercise different? And so, to say that you need you need a bunch of measures that you can try to compare, and you'll find that some of them are informative, others maybe not. Right. Sure. Yeah. I mean, this is still relatively detailed because we're looking at small sequences. Yeah. Mm-hmm. Yeah, it's on.
Ask, have there been any extensions of these two specific models where assumptions are made about landscapes and perhaps like stronger sufficient conditions are derived? Here, for example, I mean, when you say if all instances of sign epistasis are reciprocal, then there are multiple peaks.
Yeah.
This holds for all landscapes, right? Right. So, but this is also, you get very little out of it.
Yes.
I mean, if you make some maybe realistic assumptions, then could you improve upon, has that sort of thing been done?
Yes, so I mean, probably. So the next topic after discussing these empirical landscapes that I want to talk about are these probabilistic models, and of course these models define ensembles of landscapes, right, which have certain properties, and then within those ensembles, of course, these questions can be answered more precisely. On the other hand, this always assumes that the model is in some sense valid, right? So it's nice to have something that is also independent of the model. And maybe, I mean, maybe there could be further constraints that you could put on the landscapes that could lead to more precise statements, but I'm not aware of that. Yeah.
Reciprocal sign epistasis is a necessary condition for multiple peaks. if you knew how many pairs of loci show reciprocal sign epistasis, does it tell you something about the number of peaks? Seems like there should be information about that, or maybe at least.
That's a very, I mean, it's something that we're actually working on. So this is sort of, goes back to how these different things are related and to what extent you can infer global properties from local observations in some sense, right? So if you because enumerating the amount of sign epistasis is a kind of local probe. So you just look at all pairs of mutations and you count how many of them are sign epistatic. And then you would like to know, can this be, does this give you an estimate of the number of peaks? And in a statistical sense, this turns out to be true. But this is very much related to Suman's question in the sense that this is a statement that we can derive for certain classes of models. So these are models that have, a certain structure of random Gaussian interactions. And within that, we can find such a correlation. And if we then look at empirical landscapes, we also see that there's such a correlation. But of course, it's a correlation. So it's not, it's not a statement that can necessarily be applied to a single instance. Lunch?